The goal of this process is to copy a short region of DNA many times, otherwise known as amplification. First, a DNA molecule with the sequence of interest is to be denatured by heating. Once it is cool, primers are added that bond to the denatured DNA strand. Then 4 deoxyribonucleoside triphosphates and DNA polymerase are added, which allows for the synthesis of 2 new strands of DNA. This process is then repeated, doubling the amount of DNA. The repetition of this process allows for exponential increase in the number of copies of the DNA sequence. However, denaturing the DNA at such a high temperature would render the DNA polymerase non-functional. Mullis solved the problem. He figured out that a bacteria, Thermus aquaticus, could survive extreme temperatures, meaning its DNA polymerase would function. From then on, that bacteria was used in PCR. PCR helps in DNA sequencing to determine unknown amplified sequences. As far as biotechnology goes, PCR allows for the early diagnosis of malignant diseases. It also helps with the identification of non-cultivatable or slow-growing microorganisms. PCR-based tests are also able to detect the presence of pathogenic agents earlier than serologically based methods.